Evening, everyone. Thank you so much for joining this evening. This is a webinar of AONM's, the Academy of Nutritional Medicine, on Mitochondrial Magic, tips for restoring mitochondrial health. And I'm delighted to be joined by Professor Brigitte Konig, who gave a talk 2 weeks ago on the MMD tests that she does. Hello, Professor Konig. She'll be joining us at the end to help answer questions and may also during the course of the presentation help out. And I'm Gilian Crowther, Director of Research at AONM, nutritional therapist and naturopath. So we'll start off now, I think, because there's a lot to cover. So I'll go to the screen. Just need to share. There we are. So we're talking about the mitochondria this evening, and the mitochondria, as I'm sure you know from the presentation a few weeks ago and also from your own general knowledge, are the powerhouses of our cells, though they do an awful lot more as well. I have just a couple of slides to remind you what they are from a wonderful presentation. Well, it's a film actually by BioVisions, available on YouTube, called Powering Our Cells. And I think this is amazing because it's just one cell, but it's the only image I've ever seen that shows how many mitochondria there are in each cell. Most medical textbooks show just 2 or 3 floating around in the cell, but actually our cells are packed full of them. Some cells have thousands and some even millions, like the substantia nigra that produces our dopamine in the brain. And they all have electron transport chains, which are made of 5 complexes. And it's useful, I think, to remember that there's not just one electron chain complex in a mitochondria, there are thousands. So we're going to partially connect what we're presenting today with the tests that we're now AONM doing with the laboratory MMD in Germany. And I'll be talking first of all about the pathways that may have become compromised. Both macronutrients and micronutrients, of course. The macronutrients fuel access. Well, are the macronutrient fuels accessing the cell sufficiently? And are the fuels the most efficient for generating mitochondrial energy? And in terms of the micronutrients, particularly those that you need inside the mitochondria, have you got the correct substrates for the Krebs cycle, which in turn supplies the electron transport chain? And that's also called the citric acid cycle. Are the mitochondria able to metabolize those substrates? And if not, why not? And do the complexes of the electron transport chain itself have the right substrates? And of course, before that even, you've got to think about the membranes, because you have both cellular and mitochondrial membranes. Are they of the right composition? Are they intact? Are there any blockages in the membranes themselves? And in terms of oxidative stress, that's the reactive oxygen species that can particularly stop the cell from being able to metabolize oxygen, because the mitochondria recognize that the cell would be in danger if they continued doing that. Have you got high ROS within the cell? That's reactive oxygen species, or high ROS within the mitochondria? And then, do you have sufficient numbers of mitochondria in the cell? And is the composition of them in order? Are they intact? And then along the electron transport chain, you may have blocks or a proton leak. In other words, the protons that are meant to help generate ATP, that's adenosine triphosphate, the energy currency of the cell, are they leaking out through something called an uncoupling protein or through non-intact membranes? So continuing on, I'll first talk about what you should remove before talking about what you should add, similar to the functional medicine teaching that always reminds us that there's often so much to remove before adding, though I need to remind myself, that of course, without mitochondrial energy, a lot of the pathways for removing what's blocking the cell may not work. So you do have to do this in parallel. You can't just not power the mitochondria until you've resolved all the issues. But what may need attending to that's really providing or causing a huge obstacle to mitochondrial function? You may have viral or bacterial infections in the cell. These increase non-mitochondrial respiration because the cell uses oxygen to try to kill these pathogens rather than for energy. Borrelia, for example, the causative agent of Lyme disease, actually steals ATP from their host to fuel their tail with which they move through the tissues called the flagellum. Biotoxins, mycotoxins, uncouple the mitochondria, and ochratoxin A, for example, inhibits complex 2. Some parasites also inhibit the mitochondria. This is a picture of toxoplasma, which tethers and disables the mitochondria. You can actually see it tethering the mitochondria there. And heavy metals induce permeability in some cases, or increase mitochondrial reactive oxygen species formation, lipid peroxidation, and collapse the mitochondrial membrane so that it can't produce energy anymore. Pesticides and herbicides are a huge problem. For example, glyphosate, also called Roundup. This blocks the shikimate pathway in bacteria, which is the way that bacteria produce energy. But of course, we do have bacteria in our microbiome and all over our body. And if their energy production is being blocked, we don't get the substrates that they produce for our cells. Household chemicals, chemical contaminants, all important, and we'll have a couple of charts on EMFs, medications, and GI issues now, so I won't talk about them right here. Antidepressants and mood stabilizers. This is an excellent article that lists exactly what complexes these various psychiatric pharmaceuticals and other mood stabilizers block. And you can see amitriptyline comes up in a couple of, in fact, in 3 cases. Metformin blocks complex 1. How many patients do you know on that? And paracetamol, complex 3. Proton pump inhibitors bind to complexes 1, 3, and 4 and block electron transport. The reference for that is below. I think that's quite extraordinary. A lot of patients are put onto omeprazole, lansoprazole. And aspirin is both an uncoupler and inhibitor of mitochondrial electron transport. So if you're not managing to make progress with your patient, it's always worth thinking back to the medications that they're on. I actually put oxidative phosphorylation, which is the process of energy production at the end of this chain, or simply mitochondria, and the name of the medication into a search engine and have a look and see what I can find. And sometimes it's very, very worrying. So EMFs, electromagnetic frequency, disrupts the mitochondria hugely because they are actually powered by electrons. These little electrons are hopping along those complexes and producing energy at the end. Very, very sensitive to frequencies of different kinds, particularly the very powerful ones that we're now surrounded by day and night. So the voltage-gated calcium channels on our cells are sensitive to EMFs and do open up and allow excess calcium in under the influence of EMFs. And actually, articles are available to show that they allow the influx of iron as well. So actions that we can take are multiple, and there are actually companies out there that can help you with that. So particularly if you or your patients are very electrosensitive, it's probably worth speaking to somebody who's focusing on that specifically. Lipopolysaccharides from gram-negative bacteria can disable complex 4 of the electron transport chain. This is called cytopathic hypoxia. And again, another very, very interesting article that talks about that. And of course, it's not just E. coli, for example. It's something that can potentially occur with all gram-negative bacteria. You can see in this chart how very clearly they've labeled the membrane depolarization that occurs and the low ATP. So that's your gut microbiome and your stool tests that will show what's potentially going on there. Interestingly, hydrogen sulfide, which is one type of SIBO, that's small intestinal bacterial overgrowth, can cause a block at cytochrome C oxidase, which is the 4th of those complexes we were just talking about. And it is toxic to the mitochondria. So hard to pick up on a SIBO test, but worth keeping in mind. There are now increasingly ways to pick that up. And this chart here shows how that can really, really impact the cells. So what can you do to support the pathways that need work? Well, if you've seen from tests or suspect strongly that the patient has low total and or glycolytic and mitochondrial ATP, one key initiative is to try to improve nutrient access to their cells, because they may just not be getting the nutrients into their cells in the first place, let alone into the mitochondria. And just a few of the reasons for that can be uptake issues. It's worth remembering insulin resistance, as this chart shows here, the hyperglycemia that may occur in parallel with that. Digestive issues. Is the patient or are you actually breaking down your food sufficiently to go into the bloodstream and then provide the nutrients you need? Have you got the right enzymes? And you do need enzymes for carbohydrates and for proteins and for fats. Is one or several of those missing? Upper fermenting gut may be an issue. I know Dr. Myhill, who'll be speaking in a couple of weeks, will be telling us more about that. Membrane health is terribly important as well. We have 75% phospholipids in our cell membranes. And if that membrane is not intact, then we won't get proper cellular exchange. And so there are various food sources for that I've listed here. And you can also take it orally or even IV, that's intravenously. There are other reasons, of course, like having insufficient numbers of mitochondria, and perhaps Professor Konig will tell us more about that afterwards. That's something that can be measured on the tests that we now have. So going on to high anaerobic glycolysis, that means anaerobic means without oxygen, and glycolysis means making energy from glucose. So this is meant to represent the mitochondrion down here, the yellow circle. And you've got glycolysis going on in what's called the cytosol, inside the cell, but the mitochondria are not, they are swimming within the cytosol. And as we saw in that picture with which I opened, many of them, not just one. So this will mean that you've got glucose getting into the cell, but the fuels are not getting into the mitochondria sufficiently, neither glucose nor fats. This produces lots of lactic acid, and that can cause pain, like the athlete at the end of a marathon. But of course, in your case or in your patient's case, it may be just a little activity causes high lactic acid because of the recirculation in the cytosol and the inability to fire up your mitochondria with oxygen. Stress can do that, very high stress, whether physical or psychological, because the adrenal pathway gets activated, cortisol, and that fires up your release of glucose to run from a lion, metaphorically, and you're no longer using fats. And fats actually are the prime fuel for the mitochondria. If you get one molecule of glucose into your mitochondria, that will produce about 2 molecules of ATP, but I'm sorry, if it goes in, if it's only in the cytosol, it will produce 2 molecules of ATP. That's your sort of emergency energy production. In the mitochondria, it will produce ideally about 34. But if you manage to get fatty acids into your mitochondria and right through to the end, they can produce about 4 times as much. Oleic acid, for example, about 146 ATP. So imagine how much more energy you can produce if you can actually get fats into your mitochondria and along that precious electron transport chain. So other things may be pathogens within your cell. You may have viruses, bacteria, mycotoxins in there, as we've just discussed, and you may have lack of substrates. You may simply not be able to get the right nutrients into your cell, as we've just discussed, and you may have insufficient mitochondria. Again, they'll realize the power that they have and simply not accept oxygen if they know that they're not going to be able to process it. And heavy metals, of course, must not be forgotten. We'll look at them in more detail in a moment. So this is the first part of the Genova organic acids test illustration that I'm sure you're familiar with. And this is interestingly inside the cytosol, the first top bit, and you've got to get down to acetyl-CoA in order to enter the mitochondria. That's where the yellow circle begins. And you can see, in fact, there's an awful lot of information on this that we get back with every organic acids test we do. You can see that you've got arsenic, mercury, antimony blocking entry to the mitochondria. You've also got various nutrients that you must have. I've listed them up here. Magnesium, lots of B vitamins, B5, particularly important. This pantothenate here is B5, and alpha lipoic acid, carnitine for the fats, and zinc. So going on to the next chart here, this is the second part of that. When you've got inside the mitochondria, you can actually see that there are further heavy metals blocks that may prevent the nutrients from getting into the citric acid cycle, as it's called. That's fluoride, mercury, arsenic, antimony, aluminium, zinc, and there are others as well, but those are the main ones, and they're even telling you exactly at what point that can happen. And then the substrates mentioned, glutathione, a wide range of Bs again, magnesium, manganese, iron, and malic acid here. Very important. That can really help to trigger activity to get into the mitochondria too. So here you can see another chart, more detailed on the Bs. It's called biosynthesis of pantothenic acid and coenzyme A. And apart from all the Bs here, which I've listed on the right, we now have biotin listed as well, and B12. I'd just like to point out the number of times that you can see NAD, that's nicotinamide adenine dinucleotide. NAD is hugely important for getting energy into the mitochondria. That's B3. There are different forms of B3, and you can see NAD here and here and here as well. And FAD is flavin adenine nucleotide. And you'll remember riboflavin is another word for B2, and that is synthesized from B2. So it's B3 and B2 that you absolutely need, rate limiting. And the next chart reminds us of a wonderful lecture at Mitochondria Day last September, held by Biolab, where a professor from Finland who specializes in this talked about her studies on NAD+, and how that can actually improve muscle performance. Just taking that, she gave excellent information on that, though, of course, towards the end there in brief, the evidence suggests that niacin is an effective NAD+ booster in humans that improved muscle performance in mitochondrial myopathy. So from the glycolysis pathway that we've primarily been discussing just now, that goes in separately into what's called beta oxidation. And that's a perfect way to get into the mitochondria, if you can get that working. And the lactate pyruvate plus test that MMD does so you can obtain it from meat, fish, poultry, and milk, primarily. In dairy products, from the whey fat fraction, sorry, there's a spelling mistake there. There is also some in cereals and legumes, but not as much. And so vegans and vegetarians do have a disadvantage there. The next article that I've highlighted here does show how very different the pathway or the utilization of minerals is, or maybe in mitochondria compared to in the serum. And so this is actually a lovely article with lots of figures in it, detailing exactly what minerals they found in the mitochondria versus the serum in the cohort they used. And I won't go into the details of it now. I did notice that there's very, very little calcium in the mitochondria, for example, whereas there's definitely more copper, and magnesium is very important. They actually call it the metalome, which is similar, I suppose, to the microbiome and the virome. So there are special nutrients that have been put together with the right sorts, or at least approximating the right sorts of mixtures for the mitochondria. It's worth keeping an eye out as the science develops. It's very hard to get information on this exactly because the mitochondria are so tiny. We discussed last time that there are about, or you could fit about 1 billion on the head of a needle, or I should say the head of a pin. And inside those mitochondria, you have these thousands of electron transport chains. So you can imagine how very, very difficult it is, how very challenging it is for scientists to really find out what's going on in them. But the science is leaping ahead in very, very quickly. So the next slide talks about the cell danger response. It's worth remembering that this is an evolutionary conserved response, meaning it's very, very old, that a cell, when it encounters a threat that could injure or kill it, recognizes that, and the mitochondria, in our case, downregulate as a protective mechanism. So that circles back to what we talked about earlier. If the cell recognizes that it's being attacked by viruses, bacteria, fungi, parasites, chemicals, other kinds of shock, that it will downregulate its use of oxygen in order to, and that's on the next slide, these are brilliant articles by Dr. Robert Navio and his team. He only wrote his first article, which is based on substantial research. It's not just a hypothesis, in 2014. So this is all still developing science. But he calls what's going on in the cell at this point, when you have a CDR going on, oxidative shielding, not actually oxidative stress. In other words, the oxygen is leaving the mitochondrion and doing work in the cytosol instead, trying to activate viral responses to get the pathogens out of the cell or push out the heavy metals. This extract from his article, Oxidative Shielding, does explain that. And the acute cell danger response, note acute, it shouldn't go on forever, you shouldn't get this chronically, has a number of steps that have been identified, including releasing antiviral and antimicrobial chemicals, if those are what is needed, and inducing sickness behavior. If you're feeling you can't get up, you just have to lie in bed, you've got a temperature, a headache or whatever, that's all part of what the cells feel you need to do at that time, but that shouldn't continue on into a chronic response. That switch can be persistent, and that's a real problem. So let's have a look at why that can happen. You may have, and this is something that, again, the tests we talked about 2 weeks ago do show, you may have excess ATP utilization in the mitochondria, even though you're at rest, you're not even putting them under pressure. And that will likely indicate high oxidative stress. There's a marker for that, which we haven't talked about yet. We will be doing another webinar on the supplementary markers. That's called NRF2. And if that's upregulated, that does show high oxidative stress. The cell is attempting to reduce it within the cell. And also there's something called the mutant, the deletion mutant 4977, which gets upregulated. And again, Professor Konig and her team have found ways of identifying that. And that is not just genetic, that is something that can be downregulated again, but recognizing it means that that's what's going on. Now, this slide looks extremely complicated, and it is. I've referenced where all this comes from at the bottom. I just want to point out a particularly interesting and I think very insightful approach called the root cause protocol. And I don't know of many other scientific approaches that are actually focusing on ceruloplasmin, its function as a copper carrier, because without ceruloplasmin, you can't transport copper, bioavailable copper in the body. And without that functioning at these cells where iron gets trapped, you can't actually get the iron out, and you will have dysregulated iron within the cell. That's iron overload in even the mitochondria. You can see it can go into the mitochondria. The little spoon-like container of ferritin there is building up, but you're not necessarily going to see that in the serum. This is going on inside the tissues, and very complex explanations, but it's all there in the literature. Without bioavailable copper, you're not going to be able to release it through the ferroportin doorway, and you'll get all sorts of dysregulation, particularly after COVID, interestingly. COVID has led to iron dysregulation, which we did touch on a bit in the long COVID day held by Biolab a few weeks ago. So this is an article from the Weston Price Foundation. I've linked to others below, showing that retinol, interestingly, is the backbone of ceruloplasmin, and without sufficient retinol, which is a fraction of vitamin A, you won't be able to build sufficient ceruloplasmin to carry this bioavailable copper around the body and do its work. So again, even though this looks complicated, basically we're talking about a simple sequence of building the patient's retinol, making sure they have enough ceruloplasmin, ensuring that they have sufficient substrates to build that bioavailable copper, and then hopefully a lot of the ROS, these reactive oxygen species, will be quelled of their own accord, and you won't have to give the patient huge numbers of antioxidants. Many of these articles that I've quoted here do say that you can almost overdo it with antioxidants because the oxidative action in the mitochondria and in the cells is so vital as well. So there is evidence out there. This is an old article showing, I don't know when it's from, I'll have to look that up, but you can see from the style that it's probably 1965 or so, that the retinol fraction of vitamin A does build ceruloplasmin in animal tissues. And so interestingly also, mitochondrial antiviral signaling is retinol dependent. And so those antiviral signals that we saw earlier that your cell danger response is attempting to send out will have a very difficult time if that entire pathway I've just mentioned is not working. Proton leak, that's if inside the mitochondria, inside the electron transport chain, you've got a leak. Why might that be happening? Let's look at what the complexes in the electron transport chain need. Again, complex looking illustration. It's from a recent article, really, really excellent, by Vesselink and team, and gives a lot of information. You can see here the 5 complexes of the electron transport chain: 1, 2, 3, 4, and 5. And what I've done is list on the next page another illustration from the same article. I've listed the nutrients that they have highlighted in that that image, which is also explained in huge detail in the article. They've gone into every single one of these nutrients, how they've found it, the level that they've found, why it's useful, or whether the evidence isn't yet fully in yet. But they've mentioned for complex 1, B12, for example, selenium, CoQ10, vital for the first 3 complexes, 1 and 2, and then by the time it's got delivered to 3, it's not mentioned there because you need another carrier instead from 3 to 4. But you've got melatonin mentioned there. This is an article written by a Dutch group, so there it is available over the counter, whereas in the UK it's not, unfortunately. But it's interesting that there are teams across the world highlighting the antioxidant and mitochondrial boosting properties of melatonin. With the vitamin E mentioned, they're talking about the tocopherol fraction, and magnesium, of course, absolutely vital. That's rate limiting for complex 5. It should actually be called magnesium ATP. Without magnesium, you're not going to be able to fire up the last stage of your chain in the mitochondria that produces your precious ATP that's oxidative, not anaerobic. So this here shows how many copper complexes there are in the mitochondria, something that seems to not get quite enough focus in the literature. I mean, it's there, but it doesn't get talked about a lot. Copper is absolutely vital, especially for the 4th complex, which is called cytochrome C oxidase. And this actually, these little golden spheres here, and this is a complex that is mirrored by a second one. So you don't just have 4, you actually have 8. And then in cytochrome C itself, you can see up at the top, you've got 1 as well. This simply will not function without bioavailable copper. And detail in the article, very easy to access. You don't have to go to a library to access this. It's available on the internet. And interestingly, heme synthesis is correlated with cytochrome oxidase activity too, and the heme copper oxidases in most bacteria as well as in our mitochondria. So imagine how much of our metabolism, as well as the metabolism of the soil, plants growing and so on, is not going to work if you don't have sufficient copper of the right type. Sorry to be focusing on that so much, but it's a particular passion of mine because this root cause protocol really does seem to contain a lot of answers that don't receive sufficient focus. Lots and lots available. There is the link, and you know, you could be reading for years without even needing to pay any kind of subscription, and all the answers are there. In fact, Mercola just did an interview with the key proponent of the root cause protocol, Morley Robbins, who I met in Seattle several years ago, and then determined to sort of follow once I heard him speaking on the same platform as many other brilliant doctors. He was interviewed by Dr. Mercola, and his 2-hour interview is really quite stunning. So this is an article I wrote for Only Natural, the Natural Dispensaries brochure a couple of years ago on the miracle of CoQ10. I've listed a couple of articles about CoQ10 at the bottom. Of course, there's so much about it that's available to read, but what's important is that you can't generate it well endogenously if you're on a statin. So think about your patients who are on statins. They definitely need CoQ10. It's just tragic if that's forgotten, because they will not be able to generate energy like they should. There is a supplement supplier called Tisso, who we've been enthusiastic about for many years because they have dedicated themselves to the mitochondria for over 2 decades. And so I just wanted to mention them here because it's not that easy to find really pure supplements that don't have any excipients or additives in them. And sometimes that's simply what you need, or your patients need, is extracts that are really pure, because they simply cannot go out and obtain all the nutrients and cook them and then leave out the carbohydrates that they shouldn't be having excessively. It's quite a challenge. So I just want to highlight the study by Professor Konig, who's here with us on the call, into these supplements. She did this a few years ago and analyzed the capacity of some of these products and found that they had a very high capacity for mitobiogenesis. That means the generation of new mitochondria, for ATP generation, and mitochondrial metabolism. So this study is very interesting. You can see Professor Konig, MMD, up there. Mitochondrial analysis, she found, or her laboratory found, that the factor responsible for mitobiogenesis rose by up to 5,400% with some of them, and this can be measured by PGC1 alpha, which is one of the supplementary tests she offers. So on the first patient, we can see here the PGC alpha went up with the ProDLV product. That's one that contains 44 nutrients. It's like a sort of multivit, multi-mineral for the mitochondria, by over 4,000%. And prozirtuzan, which is a resveratrol product. It's made from Japanese knotweed and other synergistic components there. That was able to increase mitobiogenesis by over 3,000%. So I'll leave that for you to look at in detail if you'd like to. Just to mention a couple of the products, ProDLVit 44. The ingredients are listed below and linked to the different complexes of the electron transport chain. So I think that's interesting since we've discussed them just earlier. And there's glutathione, for example, very important, which I haven't yet mentioned, a vital antioxidant. It can really help to take that when you know that your mitochondria need a boost, or the precursors to it, which are cheaper. And some say that glutathione needs to be broken down anyway before it gets into the cells and into the mitochondria. So that can be just as useful. These are the precursors to it, and other supporters of glutathione synthesis, and others that help to resynthesize and reactivate it. So the mitochondrial triad consists, it's sort of just a term used for those three, the ProDLVit 44, the Cytosane, and then the curcumine product, because they're also helpful for different aspects of the mitochondria. This one is a synergistic combination to activate sirtuins, hence the name. And this last one I'm mentioning today is Procurmine Complete or Procurmine Forte, which has a very interesting effect on the mitochondria relating to the ability to undergo apoptosis, which is probably something we need to spend a separate sort of session on because that's quite complex. But apart from that, it's very anti-inflammatory and helps in rheumatic disorders and so on. They are giving a lecture because they focus so specifically on the mitochondria as well as the microbiome. They are giving a lecture on the 11th of June, and there are a few free places. So do, if you wish and have time, contact them here. I'll be there, and that'll be online. So that's exciting. There are lots of resources available on these orchestrators of our cells. So no dearth of literature to read if you wish to. You can see Dr. Sarah Myhill's book here. It's Mitochondria, Not Hypochondria. And she's updated that and written many more since. She's very prolific. This is a book by Professor Konig, who's written many and many, I mean hundreds literally of scientific articles, peer-reviewed. Just to mention that Sarah, Dr. Myhill, will be speaking on how is mitochondrial dysfunction linked to chronic disease and what can we do about it on the 12th of May. That's Awareness Day. It's actually dedicated to ME worldwide. So please do join us for that at 6 o'clock. And thank you very much. Brigitte, I don't know if you want to turn on your view again. Lovely, thank you. I don't know if any questions have come in. I think I might have forgotten to say, please put your questions into the chat box or the Q&A box, but there's time for that now. Have any come in, Brigitte?
Only a few.
That's all right. My fault, I forgot to say it, but I'm sure others will come.
There was one question, is iron release connected to hemochromatosis? Hemochromatosis is a genetic disease, so it's different from mitochondrial dysfunction. Sure, it might be lead to mitochondrial dysfunction, but hemochromatosis is still really a genetic disease. Then there was a question, where it is. Oh, now are coming some questions. No, only very thank you. No questions, no questions, no questions.
Well, I saw one just, go ahead.
There was one zinc was listed as a heavy metal that acts as a mitochondrial blocker. Is this referring to very high levels of zinc supplementation? Yes, zinc is a heavy metal, but you need it in very, very low amounts. Yeah, and like every element, if you take it too much in too high concentration, it's bad effect. Yeah, but it's always the same with all things. But there are also some heavy metals still in very low concentrations. They are not good, like mercury and arsenic, and there you should avoid every small amount of this heavy metal. Yeah, so there are some heavy metals you need in low concentrations, and there are heavy metals you should never take them. Yeah, or yeah.
Yes, or chelate if you find you have them. One question in the Q&A is it relevant to mitochondria if ferritin is low and folate is high? What would you say there? That's B9, if folate is high.
Sure.
Go ahead, sorry.
No.
Well, I was just going to say that having low ferritin, if it's measured in the serum and there is no other way to measure it, doesn't necessarily mean that you don't have ferritin locked in the cells, as we've just seen. Something quite new for me as well to begin to understand that. But so I wouldn't say that you don't necessarily have reactive oxygen species just because you have low ferritin. Would you agree with that? There are other markers of ROS, aren't there, and not just ferritin in the serum. But with folate, I can't say I know of a correlation there.
No, I don't know.
It depends on the illness you have, doesn't it, and how you're fighting it. Sometimes it isn't at all a good idea to have too high a level of folate. One question in the Q&A is also, is it possible to buy the supplements if I'm not working with a practitioner? Those ones that I just mentioned in the presentation, yes, they're available at the Natural Dispensary. And that's a wholesaler in the UK from which anybody can purchase, as far as I know. What does a high copper result on a lab test mean? Would you like to address that? Or is that something?
No, it depends which lab test. Yeah, it's in blood. It is after giving some MSN or whatever. Yeah, it depends which high copper result, where and when.
Okay. One doctor here asked about the tests. He said, can you tell us a little bit more about the tests and how they function? There was a presentation by Professor Konig a couple of weeks ago that's now on the website. So I've noted your name and I'll send you a link to that. I think it's probably more useful. I mean, it's basically using the Seahorse XF, isn't it? The MHI, the Mitochondrial Health Index, but it's a combination of different methodologies. So it's probably more useful to watch that first, and then she'll be giving us another presentation about the supplementary markers soon, and then we can ask her additional questions that come up that she didn't address in that first presentation. Yes, there was another one. Please do elaborate on the tests that can reflect mitochondrial function. They are available on the AONM website under mitochondrial tests, and there's quite a lot of information there about them, but obviously we're very happy to address further questions. There are some samples of the results there, and I'm sure Professor Konig is always happy to have a conversation with somebody who'd like to understand more than is there. You've done that already, haven't you, Brigitte? You're happy to talk to individuals or small groups about testing.
Sure.
It's something you're very passionate about, as I am really. And otherwise, I'm looking for, yes, is there a good heavy metal detox that Professor Konig can suggest to help our mitochondria?
Yeah.
It depends a bit on the metal, doesn't it?
Yes, it depends on the metal. Sure, you can do a chelate therapy. You can take to prevent that the gut absorbs a lot of heavy metals. That is one way. But first you have to remove it from your body. There is different, really, the chelate therapy should be done, but it depends really on the heavy metal, and you should talk with that about your therapeutic, or if you are a doctor. I think, Gillian, you have some specific seminars for chelate therapy.
Chelation therapy is difficult in the UK. We can talk about that separately. It's not well recognized, but there are definitely people who do it using pharmaceuticals like EDTA and DMSA. From what I understand, I know that's used much more frequently in Germany, but there are also natural ways of detoxing, aren't there? Again, depending on how heavy the load is and what it is, and where it is, and whether it's dental or appears to be in the brain or whatever. It's something, as you say, best to discuss with your therapist. I'm just going backwards in the questions to see if there's anything else. Would you like to mention anything otherwise specifically? What herbal medicines are good is a question here. I think that's too sort of big a topic to go into, because it depends so much on whether you're talking about addressing infections or detox of heavy metals or supporting the mitochondria. And as I hope I made clear, and I think Professor Konig really goes along with me on that as well, that it's not always about just pushing good nutrients into the mitochondria, depending on what else is going on. You'd agree with that, wouldn't you, Professor Konig? About supporting the system as a whole. And if you've got these blocks, as it were, what I mentioned under removes, you do need to think about those hard as well, don't you?
Yes, it's not so easy. It's not just to take a few tablets and then everything is going well. So you have to decide, it is really a problem of nutrients. Do you have enough of nutrients? But sometimes the people have enough of nutrients, or they are taking for years a lot of nutrients, but it doesn't help. So they have it in their serum, but it's not going into the cell. It's not working. And also you must think which way is the best way. Do you have mitochondria? Or perhaps your cell has a lot of mitochondria because they are producing a lot of mitochondria, compensating for the deficit in ATP. But then it's not good to do interventions to increase the mitochondrial number again, because then you will increase very deficient mitochondria. Then you have to go into mitophagy to delete the wrong mitochondria and then multiply the good mitochondria. So it's really different, and therefore we have, unfortunately for you, a complex test. We can't say one test or one ATP is telling you everything. It's unfortunately not functioning. Therefore you have we have the mitochondrial health index with supplementary markers to see exactly where to go. And so it must be decided individually.
Well, I know you've said kindly that you're prepared to speak to therapists once a test has been done to help interpret it. And I think because it's stunning that you can look at the individual complexes of the electron transport chain and see what's going on with each one of them and where there may be blocks. That's not a test we've introduced yet, but I do hope that eventually we can.
Yes, we can do it with that is a classical biochemical test. We have it. We can do it in a routine. And if I just have the blood, I see a lot of components. But I would like, if they like the therapeutics, I can talk with the practitioners, with doctors, because they have more information about the patient. Yeah, is he already taking supplements for years, or what are additional parameters? Yeah, and then they can tell it to me, and then we can plan together. Yeah.
Yeah, I know you've told me that you can do cell-free DNA.
Yes.
Which is very helpful, and that you can even identify whether it's mitochondrial
Yes.
or nuclear DNA if necessary. But there's a question here, can you measure the efficiency of the translocator proteins that are actually translocating nutrients from outside into the mitochondria and the other way?
Yes.
That's something you can do?
Yes, but that is we didn't talk about it. Yes, yes, we can do it. We have a special test. Sorry, Gillian, we didn't talk about it. We did it yeah, since several years. So you can work with specific media, and we exactly know then whether the translocation is from glucose or fatty acids or ketone bodies or amino acids. Yes, we can do it. Yes.
Fantastic. Thank you. Well, we must look into that as well. And I know you've also talked about the use of, for example, C8 in a beta hydroxybutyrate to go straight into the mitochondria if there is a block at pyruvate dehydrogenase, the complex at the beginning of the Krebs cycle. There is a way of sort of leapfrogging over that.
Yes.
And that can really help, can't it, to reactivate that process?
Yes, yes, we had several patients where we could see that that it would be beneficial, but we have also patients it would be do nothing. But yes, we can exactly test it and analyze it. Yes.
What I haven't talked about today, and we haven't gone into yet, is the other sorts of non-nutritional, nutraceutical ways of activating the mitochondria. And of course, because they're little sort of electric beings, there are many ways as well, like near infrared light and far infrared light and so on. And we'll definitely be covering that. I mean, that probably won't be me. We'll get specialists to talk about that. But there are lots and lots of ways, aren't there? Autophagy, perhaps one of the last questions here. Mitophagy, what are the ways you know to actually trigger that if you can see that the mitochondria are damaged and need to be replaced?
Sorry.
You talked about mitophagy. What are the ways that you know of that can trigger that? I know that intermittent fasting, for example, is a good way, isn't it?
Yes, intermittent fasting or always fasting, but also specific kinds of sport. Not too much. It's yes. And mitophagy is also induced by intermittent hypoxia. You can use it, ERT.
I know that that's available in the UK as well. Right. Yeah. A few more questions in the Q&A.
Okay.
Do you need to treat pathogens before dealing with the mitochondria?
Sometimes you have to do both. It depends on the patient. If he is very tired, you should go on both things. But you should treat the pathogens. Sure, only to supplement the mitochondria is not good. Yeah, but you can go dealing with intracellular bacteria plus supporting the mitochondria, so the mitochondria can work better. Yeah,
I know that's what Dr. Klinghardt and a lot of other therapists say, is that you may possibly be feeding the bacteria, particularly in the gut microbiome, if you're giving too many goodies all at once at the beginning. But being targeted about it and keeping the mitochondria going is, of course, very important. Another question here, that's sort of more a different kind of topic. If I have unconfirmed, but very probably chronic Lyme, is it still worth taking some of these nutritional supports? I think that falls into the realm of what Professor Konig has just said. Otherwise, one would probably need a bit more of a consultation to answer that.
Yeah.
just going through the rest of the, now I think that's most of the Q&As. There is obviously a lot more in the chat. People are asking, will a recording be available? Yes, it takes a few days and then we put it up on the website, and the PowerPoint, the charts will also be available, yes. So just to mention again, thank you so much, Professor Konig, for responding to those questions. We'll be having you very soon to talk about the supplementary markers, and that'll be exciting again. Let's just mention what they are, because we didn't even say what they are on the last, at the last webinar. They are PGC1 alpha, NRF2, and
the number of mitochondria.
Yes, lactate pyruvate plus, showing the kinds of nutrients that are able to access the mitochondria, and the 4977 deletion mutant that can be reduced again, not purely genetic, but shows reactive oxygen stress in the cell, in the mitochondria particularly, and NRF2.
Yes, anti-defense, antioxidative stress response.
Yeah. So I think those are the ones we have so far, but hopefully we'll be able to introduce the others that you've already mentioned in time. And so lovely to have a talk about those quite soon. And I just wanted to mention Dr. Sarah Myhill's talk again on the 12th of May, ME Day, and very much looking forward to that. And hopefully perhaps you'll join us for that as well. That would be very, very nice. Thank you.
Sure.
So it is 8 o'clock now, so don't want to keep you too long. And I know it's an hour later in Germany as well. So thank you so much, everybody, for being there with us this evening. And if you'd like further details about the tests, then please do look at our website or simply call the helpline a call, give the helpline a call. And in terms of the answers to the different aspects of the tests, that what you can actually do, we'll be building literature on that. We definitely will. Professor Konig actually does have a sort of second set to her tests, which is suggesting what can be done, a little bit like the OAT test, you know, where you get a first page with sort of give this, give that, give everything else, but it's a little bit too 101, isn't it? It's a little bit too simplistic sometimes to do that, as you said, without knowing more about the patient. So hopefully we'll find a middle way.
Yes, that's right.
So thank you very, very much, everybody. And look forward to seeing you again. Bye.
Bye-bye.
Bye.
Good night.